So can you see my screen?
Yes, we can see. Great.
Perfect. So thank you again, all of you, for joining. And our session today is, so our session today is going to be talking about molecular dynamic simulations and using this methodology for purposes of docking. So the conventional notion around docking is that you take a protein system and you try and expose the binding site of the protein to certain drugs. I'm going to talk about this in a bit more detail as we go on in today's session. So when we are trying to expose a particular protein and its active site to a particular ligand which can act as a drug, what we are trying to do is to modify the behavior of the protein. And one way that we believe and how to do that is either to take the binding pocket or any other allosteric pockets and try and occupy those pockets with certain drugs or drug-like molecules which can then create modifications to the dynamics of the protein structure. Now, a common word that people hear is a ligand or a drug. Now, both of these at the heart of it mean the same thing. So any small molecule which goes and binds to a particular protein can be referred to as a ligand, right? But if it is actually there to modify the properties of the to modify the function of the protein, then we will call it a drug because it has been introduced specifically for the purposes of modifying the behavior of a protein which is implicated in a particular disease. Now, the conventional method of docking involves something like autodock, for example. You might have heard of this name before. Today's session is not about that. There are certain tools that have been developed, kind of like autodock. And what they do is that they sort of create the docking, sort of convert docking into a high throughput process. So what does that mean? So high throughput just means that you can screen hundreds of thousands of libraries of hundreds of thousands of molecules against your protein. Now that's all good, but the problem is that if the proteins are thermodynamic molecules, so that means that if you take up a protein structure and you put it at around 37 degrees, the protein is not going to be static. Because of the heat content of the system, the protein is going to be moving around a lot. And as a consequence of the protein moving around a lot, there's a lot of inherent flexibility. And what high throughput tools like Autodock do is that they tend to sort of approximate those flexible motions. Now, the problem with approximating is that if you are trying to make a very resolved process into a high throughput process, you're eventually going to end up compromising on a lot of things, right? So when you remove the flexibility part of the protein and you look at proteins as rigid structures, you tend to speed up your computational calculations. Molecular dynamics is the opposite of that. In molecular dynamics simulations, what you do is you try to observe the system in a lot of detail. And when you try to look at the system in a lot of detail, what ends up happening is that obviously you spend a lot more time, but because you're looking at your system in a lot of detail, you tend to capture the system as it would be behaving in reality. You will see that Autodock has been around for a long time, and a lot of other tools have been around for a long time, but we have not developed a mind-blowing number of drug molecules, right? So you will notice that a lot of things that work very well in computational predictions, when you actually take them and translate them into clinical settings or in lab settings, what happens is that they don't perform as they are supposed to. So today's discussion is how docking will work when you're doing molecular dynamic simulations. Now, the important thing to remember in this entire exercise is that molecular dynamic simulations are a methodology, and think of this as a lab in its own. So when you're doing molecular dynamic simulations, you are going to set up your experiments in many different ways, right? So today's session, we don't have enough time, so we can't cover all of those settings. But what I will show you is a very basic theoretical, I'll give you a very basic theoretical foundation on what are MD simulations, how do you run them, and well, not exactly how do you run them, that would be a part of another workshop in this course, but I would be showing you just like the theoretical basics of it. And then towards the end, we will touch on docking and how that is done. But before we start and go into the details of all of that, let's revisit some of the basic principles. So at the heart of it, you have the DNA. The DNA comprises of nucleotides. Now, these nucleotides are then converted into mRNAs, and those mRNAs then are processed, and then after processing, they actually end up at ribosomal sites where there are other molecular components which end up synthesizing your amino acid chain, right? So when you have your amino acid chain, what you essentially are looking at is a collection of the 20 natural or some modified amino acids, and these amino acids then come together and are linked in the ribosome using a polypeptide link, which connects the C terminal of one amino acid with the N terminal of the incoming amino acid. So this way you grow your amino acid chain. Now, once the chain has been produced, what will end up happening is that you will have a linear chain. Now, this linear chain obviously is comprising amino acids, and these amino acids have side chains, and those side chains will then start to interact and give rise to more complex three-dimensional structures. Now, it is important to bear in mind that there are stages. The biology textbooks usually classifies the structure in stages. So you have secondary structure components, which contains beta sheets and alpha helices, and these arise because of backbone interactions. Now, at a higher level of detail, the side chains are also shown to the left part of your screen. And in here, the side chains from different regions of the amino acid chain will come together in the three-dimensional space, and because of their chemical natures, they will, like in case of cysteines, you can have a disulfide bridge, or in case of positively and negatively charged amino acids, you might have relative ionic bonds or hydrogen bonds that tend to form up. And similarly, you have these different kinds of stacking or packing interactions, which will lead to more complex structures. As a consequence of this, what will end up happening is that the protein will tend to fold up into a more complex three-dimensional structure. Now, why is it important to remember this? Is because it is the final three-dimensional structure that will carry out whatever function is needed from the protein, right? So if the protein is not able to reach its final structure, sorry, if the protein is not able to reach its final structure, it will not be able to carry out its required function. Now, it is important to remember here that what are the things that can stop a protein from reaching its final three-dimensional structure. Now, a protein can have mutations, and mutations might change the way a protein folds or takes up its final structure. Now, I believe it is a sickle cell anemia, which some of you might be familiar with, where you have a change in one amino acid, and that leads to a change in the way the hemoglobin protein in humans works, and the cells become sickle shaped because of the mutated protein, and people become anemic. Now, when we are talking about this, it is important that a lot of proteins, when you will work with them for docking purposes, do not have their structures known. So what people usually do is they use homology to first predict the structure, and then based on that homology predicted structure, they will do docking using autodock or using whatever. Right now, the problem with that is that that might work in 1% of the cases, or maybe 2% of the cases. But underlying the principle of homology is that proteins that share similar sequences will end up producing the same three-dimensional structure. Now, the problem with that is that that might be true, but we have both extremes. We have cases where the proteins can be almost 80 to 90% having similar content, but they might end up folding in completely different three-dimensional states. On the other end, you may have proteins which share as little as 20-30% similarity, and they still end up taking the same three-dimensional structure. Now, so the reason why I'm highlighting this is that you have 2 degrees of uncertainty. First, you're making a prediction that your three-dimensional structure is homologous to a certain protein when it might not be. The second is that based on that homology, you do not have any experimental evidence. You are making a computational prediction and then docking on that computational prediction, and that is twice the amount of uncertainty that you might have. So it is always good to have an experimental based starting point. So always try to ensure that the structure that you're working with when you do MDs, when you do any kind of computational predictions, be it conformational states, be it drugs, be it function, you should always think about starting with a structure that is experimentally determined, right? On top of that, the binding pocket of a protein, if you have the protein in its native state, for example, like in its wild type states, which means that you have not homology modeled, you have actually determined the three-dimensional structure of a particular protein. Now, in that case, the binding pocket might be exactly known because you can crystallize a protein structure with the drug bound. So if the drug is bound, so if the ligand is bound to a particular protein, you will know where it binds, and that way you can determine the binding pocket, right? But what will end up happening is that if you try to predict the binding pocket, there is a high chance that the prediction might be wrong. So docking is not about predicting the binding site. Docking is always about knowing where the binding site is and then finding different drug molecules that will fit that same drug binding pocket, right? Now, there are tools which actually do this. There are tools that actually predict the binding pockets, but we're not going to go debate about their accuracies. Obviously, not all of them function in all cases. Some of them might work very well on certain classes of proteins that are conserved, and some of them might fail altogether. So there is a very wide degree of results that you will get when using these tools. And it is always important to remember what are the strengths and weaknesses of each of the methods that you have used, and then explore your systems accordingly. So once the protein structure folds, you may have the case where you have something like where one protein structure comes together with a couple of other protein structures, and this entire, so in this case, you're looking at hemoglobin, which is a tetramer. So it comes together from 4 units with 4 units. And now this state is called the quaternary structure. Now, it is important when you're doing molecular dynamic simulations or docking studies of anything to remember that the dynamics of the protein alone and the dynamics of the protein. Sorry? Was there a question?
No. Just
Okay.
some participant do not mute
Oh,
the.
okay. Okay. No worries. So it is always important to remember that the dynamics of the protein might not be identical when the protein is alone. That means that protein is it in its tertiary structure, or whether it's or if it's in its quaternary structure. So the dynamics might be completely different, right? So it is important to remember that when you are trying to discover new drugs, to keep this in mind, that will the drug function properly when it is in its tertiary structure, or whether it's in its quaternary structure. So this is always this is also something to bear in mind when you do your docking and try to find out uh, which drug is best suited for a particular interaction with a protein. Now let's come. So after this, fairly quick and very rough introduction, let's get down to what molecular dynamic simulation is, right? So in the world of computational simulations of biological molecules, you usually have to actually, we can call it 3 levels of detail. So the middle level of detail is where you take a protein structure and represent, have an atomic level representation. So an atomic level representation means that when you are creating the protein structure inside your computer, what you are doing is that you are representing each atom by itself, right? So this basically means that so this basically means that you will represent each nitrogen, each carbon, each sulfur, each oxygen, each nitrogen as a rigid sphere, right? So this is called an atomic level description, when each atom is represented as one ball, right? Now, I told you that this is the middle level. There is a level above that, which is called coarse grained levels. And in coarse grained levels, what you end up doing is that you end up packing multiple atoms together to make larger spheres, which represent a group of atoms, right? So atomic representation was in the middle. The one above that was coarse grained. Now there is one below that as well. Now, when you're doing molecular dynamics simulations, you cannot go below atomic level representation, right? So what is below atomic level? So if you recall your high school chemistry, you will remember that there is no such thing as a bond, right? We represent a covalent bond as a single covalent bond, or a double covalent bond, or a triple covalent bond. But in reality, there is no such thing as bonds. What we usually do have is something called electronic clouds or hybridization states. So you would have read this in high school, where you talk about sp hybridization, or sp2 or sp3 hybridization states. Now, so those descriptions fall into the quantum level. Now, there are other tools available where instead of doing molecular simulations using atomic level description, you can actually use the quantum level description of your system. But those systems cannot simulate entire proteins, because when you go down to the quantum level, what ends up happening is that the amount of things that you're keeping a track of start to combinatorially explode. And the problem with that is that you don't have enough compute power to actually look at an entire protein at the level of electronic clouds. So what we do is that we say that, okay, atomic level description is a reasonable compromise, or anything that is coarse grained. So atoms are considered as rigid spheres, and the reason why this is popular, that even though these are very strong assumptions that atoms act as rigid spheres, but the good thing about it is that when we try and run these simulations, and when we match them to actual experiments, there is a very high degree of agreement. So what we see in a computational world is what is seen in the experimental lab as well. So now, before we go into the depth of molecular dynamics, let's have a look at why molecular dynamics is done and what it is and what our end result would look like. So when we talk about protein structures, we can look at protein structures in very different ways when we are looking at them in a computer. So in this case, there's a protein called P24. And this P24 protein has 6 units, right? You can't see the units separately over here, but if you draw this same protein using a different representation style, you will see that now you can begin to point out that, oh, look, these are the 6 individual units. Now, why is it important to create this structure? Because once you start looking at the structural details, you can get higher level of details or how the protein carries out its function. So for example, this particular protein is responsible for making the HIV capsid. So the HIV capsid, so HIV is the virus, and that virus is packed, it has a protein coat, and that protein coat would make it look like this. Now, once we have the structural details with us, what we can start seeing is that how does one hexameric unit fit right close to the next hexameric unit. And why is this important? Because now you can start to say that, okay, if the HIV gets a mutation in the P24 protein, maybe that mutation is at the interface, and that interface region is then binding to another protein on this side, for example, and it might get disrupted, right? So if the virus's capsid is packed poorly, what will end up happening is that you will end up packaging the virus loosely, and the virus will lose its infectivity, right? So similarly, when we talk about packing, in this case of the virus, docking is a very loose term. Docking is usually thought about when you say that, oh, I want to find a drug molecule which goes and binds to a binding pocket of a particular protein, right? But a lot of times proteins don't have binding pockets. They have something called binding interfaces, which means that you won't have a clear-cut pocket-shaped thing, but you will have surface lining amino acids which can interact very nicely with something else as well. Now, as we go forward in time, what is happening is that we are looking at not just drug molecules, but peptides as well. Now, when you talk about peptides, you are looking at 6, 7, 8 amino acid long protein chains. And they won't nicely fit into a binding pocket, but they will interact with a group of residues. Now, looking at the picture that I'm showing you on the screen right now, if this unit and this unit are interacting, there is an interface between them. And that interface has amino acids on each of these 2 units, which are holding each of them together. Now, when you talk about docking, try to expand and see that docking can also be done on protein-protein interfaces. I mean, it won't be called exactly docking then, but protein-protein interface interactions are also important, because then you can study whether 2 proteins will come together and bind or not, right? And that can help us identify protein-peptide interactions, right? So that is a new class of drug molecules which are being explored at this time. So let's have a quick look at what molecular dynamic simulation would look like. So here, this is not my simulation. I just got it off of YouTube. But what it is showing is that you have ATP synthase. So ATP synthase is a molecule which is used to develop, not used to develop, it sits in the middle of a membrane, and there is the electron transport chain right before it, and that electron transport chain creates a proton gradient. Now, there are more protons on one side of the membrane than on the other, And what the protons end up doing is that they end up flowing through this green protein. They end up flowing through this green protein, right? So when they go through it, it drives the protein into a circular motion, and that then affects the surrounding proteins, which then allow for the ADP molecule to bind to a phosphate group and generate ATP molecules, right? So this is also referred to as the molecular machine. But the reason for me showing you this is that when you talk about molecular dynamic simulations, this is the kind of end result that you are hoping. Okay, so so this is one example of how a molecular dynamic simulation would look like. But you can also study molecular dynamic simulations for looking at how drugs bind. Now, when I say how drugs are binding, that is a different question from the conventional definition of docking. Now, when we talk about docking on its own, what we are saying is that, hey, here's the binding pocket, here is a drug. Does the drug fit into the binding pocket or not? Right? That is the question that you're asking when you study classical docking. But when you study drug binding using molecular dynamics, or the way I am explaining docking to you, what you will end up doing is that you will see that the protein structure has a breathing motion. So this is also something called, so what you're looking at on the screen is also something called induced fitting. So induced fitting basically means that in the presence of the drug, the protein will change its structure slightly and bind to the particular drug. So this pocket might not be visible when you start the simulation, but will be created once the drug actually reaches wherever it is supposed to bind on the protein. Now, these kinds of simulations are incredibly computationally expensive. So you don't expect them to do them in like one day or one week, but these simulations usually last over months on supercomputers, right? But the point that I'm trying to make is that they are doable and they can be done. And this helps you explore certain questions. That if you have drug A, for example, and drug A works in a certain way, and you know that way, then when you want to test drug B, you can make the protein exposed to that particular drug and see if you can, because of you already know how drug A works, right? So what you are looking for are analogs between whether drug B works the same way as drug A or not. Okay, so then in that case, what you will do is that you will have a look at the dynamics or the binding pathway, right? But to sum it all off, docking is where you have something like this, for example. So you start off with a ligand library. So this would be basically mean that you will have a directory with hundreds of thousands of molecules. And then you will do some pre-processing on them to get them in the right states. Once they are in the right states, this cloud-like representation, gray on the outside, is basically the surface of a protein, which has this cavity on it. And what you are doing is that you are picking up each of these amino acids and you are throwing it into the binding pocket, right? And every time you throw it, you calculate the interaction profile between the drug and the binding pocket. And you say, you go through all of these, and then you find this blue one. And you say, oh, look, this interacts better because these yellow bonds are forming. And then based on this scoring scheme that you develop, you rank your findings based on some score, right? And I'm going to tell you a few ways in which those scores are calculated. So before we do that, let's have a look at, we're living through a pandemic right now, right? So everybody knows that there's something called SARS-CoV-2 that is running around. So this particular image, it's not mine. I think I took it from a paper. Shows a particular capsid of the virus. which has the spike protein coming off from it. And what we want to do is we want to develop models where we can study how does the spike protein from the coronavirus actually interact with humans. Okay, so what would we do? So what people have done, so for example, in this particular case, this pink patch is a membrane, and then you have these components of the spike protein with the receptor binding domain at the top here, right? Now what you can do is you can conduct simulations of this. So what would a molecular dynamic simulation of the spike protein look like? One kind of simulation will be this. So you see at the bottom over here are lipids, which are holding the spike protein. And this spike protein, obviously, there is no, so each coronavirus comes with several spike proteins, right? So in this particular simulation, what you're trying to look for is how do spike proteins interact with one another, right? So because they're not rigidly packed in the membrane, right? They have certain degree of freedom. So two spike proteins might come together, and when they do come together, they will interact, and you want to study what their interaction would look like. Another thing that can be done is that you can see that, hey, I saw this as my receptor binding domain, which I showed you a few slides ago. And this blue-green protein is now from the human counterpart. And what you want to see is how does the coronavirus interact, or the spike protein from the coronavirus interact with the human protein. Now, when you do that, your simulation will look something like this. Again, not mine. This animation I got off from YouTube as well. So you will notice that I'm going to speed forward this simulation. So you will notice that the bottom part is the spike protein. The top golden part is the human protein. These green chains that are coming off of the protein are glycosylations. So glycosylations are basically modifications to amino acids where certain sugars will come and get attached to the amino acids. And this is what helps the virus escape the immune response from humans, because it presents a signature of sugar, which might mimic those of our own cells as well. So that is how the virus escapes. But what you are actually doing is that underneath those surfaces, this pink portion is interacting with this top portion of the human ACE2. And here you will see that the pink portion has certain amino acids which are at the interface and interacting with ACE2. Now, when we talk about the coronavirus, for example, we talk about different variants. So one of the things that varies between the coronaviruses is actually that there is going to be a mutation at this interface, and that mutation is actually going to make it bind strongly or poorly to this ACE2. So if the mutation comes in and the binding becomes poorer, then the virus is not as infective or as dangerous as, it won't be as dangerous, right? But if a mutation makes this interaction much stronger, then the virus will become more infective, right? So now again, where does docking come into all of this? So again, I told you about protein-protein interactions, right? So this ACE2 is one protein from the humans. This pink is one protein from the virus, and these two proteins are interacting. If these two proteins do not interact, then there is no way that the virus can enter our cells, or the virus genetic material can enter our cells. So it is very important that when you are talking about docking, so this is, I know this is not the correct example, but what I want to illustrate is that the power of molecular dynamic simulations is that when two things come together, you can see how strong or how weak their binding is, right? So the question that might be coming to your mind now is something that we're going to deal with right now, is that how do I run this simulation? What is actually happening at the ground level? How are these simulations happening, right? Now, before you understand how simulations work, remember that we talked about molecular dynamics being atomic level representations, right? So when you have two atoms that are in close proximity, what you can think about is that in this particular case, this solid gray line is basically a bond which is shrinking and expanding, or relaxing and contracting, right? And when you talk about this bond stretching, you talk about degrees of freedom, right? Now, there are several degrees of freedom when you are talking about protein structures. So, for example, in case of two atoms, what you can do is you can think about it as a spring and model the amount of energy as a harmonic potential, right? So what does this mean? You will remember from your high school days that there is something called the equation of a spring, right? So what you can do is you can take that equation of the spring and modify it slightly to represent the amount of interaction between these two atoms based on this bond, right? And then you have this bond and this bond and this bond, and you have a lot of other bonds. So there is a summation sign because you're going to do this for all bonds in your system, and you're going to sum all of them up. Now, when you sum all of them up, you will come with something called the potential energy of all the bonds in your system. Now, bonds are 1 degree of freedom, right? Okay, I'm using the term degree of freedom loosely. So let me correct that. So what I mean by that is that bonds is one way in which things are moving, which you can capture using mathematical models. Another thing is that you may have motion like this. So when you have motion like this, you do not have bond stretching, but you do have something like this angle that is changing continuously. So you can again have the same kind of representation that you had before, but instead of measuring the distance, you are now measuring the angle. So there is an angle between these two, but there is also an angle between these two and between these two. So you take all of the angles in your potential, in your possible system, and you calculate them and you sum them up, and then you come up with something called the potential energy contributed by all the angles. Now, these two are very simple to understand, but there are more complex motions. So for example, in this case, what is happening is that you have something called dihedral angles, right? So dihedral are planar angles. So imagine that this bond B2 is rotating. So when this B2 rotates, this acts in a fan-like path. And this is because at this point B, there is a certain degree of rotation, right? So what we do is that we consider this as another way in which motion can happen, but this is represented in a slightly more complex mathematical notation. We're not going to go into the depth of that, but again, by summing up all of these motions, you will come up with the potential of all the dihedrals, right? So what have you done? So up till now, you have looked at length, you have looked at angles, and you have looked at dihedrals, right? So these three and some others which I haven't covered are basically called bonded potentials. They're called bonded because they are between directly either atoms that are connected by a single bond, in which case they're called 1-2 interactions, or they are angles in which they are atom 1, atom 2, atom 3, or 1-2-3 interactions, or they are dihedrals. So basically atom 1, atom 2, atom 3, atom 4. So they are 4 interactions, right? Now, if you talk about 1-5, 1-5 are not linked using bonded potentials. Anything that is more than 3 atoms away from a particular atom falls under the non-bonded potentials. So non-bonded potentials are where you have gone further out. So for example, how will amino acid 1 interact with amino acid 89, right? So again, these are basically just looking at charges. We don't have to go into the mathematical details of this, but there are partial charges on certain atoms depending on their states. And what we end up doing is that we end up looking at those partial charges or partial states. And there's also something called packing interactions, which are approximated by Leonard-Jones potentials. So these two basically, so you have the Leonard-Jones interactions and you have the Coulombic approximated electrostatic potential, and these two are classified as non-bonded potentials. So now what have you done? So you have taken 1, 2, 3 from here, and you have taken 2 from here. So this term and this term. So together, these 5, when you sum all of them up, so bonds, angles, dihedrals, so the 3 bonded and the 1 non-bonded. So if you add all of them up, you get the total potential energy of your system, right? So now this is very important to understand, because when you talk about docking, or when you talk about molecular simulations, and you're looking at how strongly 2 things are binding, at the heart of it, there are going to be several different kinds of calculations that you do, right? And obviously today, because we're limited by time, we don't have time to cover all of them. But what you are basically doing is that you are looking at how things are interacting. Now you might start thinking about, hey, but there is something called a hydrogen bond. We have not looked at hydrogen bonds, right? So I would like to remind you that hydrogen bonds are caused by charges, right? So you have an acceptor and a donor. So when you have an acceptor and a donor, what you are basically doing is that you are going to approximate your hydrogen bonds using the electrostatic potentials, right? So basically, all of those things that you can think of which are happening inside a protein. So how do ligands bind to a protein? They don't make covalent bonds, right? They just go there, and because there is some hydrogen bond interaction, they actually like sitting there because there are hydrogen bonds that are formed, right? So basically, what you can do is, by looking at non-bonded potentials, you can actually approximate how strong a particular interaction of a ligand with a particular protein is. Or if you are talking about protein-peptide interfaces or protein-protein interfaces, you can also use non-bonded potentials to actually explore those. So now the next question should be, where do all of these values come from, right? So we're not going to go into the details of that. There are people who are going to be teaching you about molecular dynamics. Maybe they are covering this in more detail, or you can ask them. But there are PDB files which are available for structures. They give you the geometrical details. So the geometry values that are recovered from the PDB file are actually shown by red arrows. I've shown them by red arrows, right? The other terms, which are shown by blue arrows, they come from whichever molecular dynamics program you're using. So if you are using, for example, CHARM, you will end up using CHARM force fields, right? Now, force field, we're not going to go into the depth of that, but the force fields are basically going to give you certain values, which you are then going to take out and plug into all of this. And during your simulation, what you will be doing is that you will be calculating, you can calculate this particular energy function for all protein states. Now, this is for CHARM, and it might be different for, it might be slightly different or very different for other programs as well. But at the heart of it, what are you doing? You're looking at how many atoms there are in your system, how are they arranged, how can they be linked to one another. So there will be bonded potentials and there will be non-bonded potentials. Now, there might be slight differences in the way the math is calculated to approximate those non-bonded calculations, but every program will be doing it. They might be doing it in slightly different ways, but they will do it, right? So these are important things to remember. So what actually happens is, during the course of the MD simulation, is that you will take up a particular PDB file. It will give you the atomic coordinates. You don't have to understand this in too much detail. I know this is an overwhelming diagram, But what you're basically doing is that you're solving the Newtonian equations, right? And those Newtonian equations, like this is just a complex representation of F equals ma, which is that force equals acceleration times the mass that is producing it. So basically you are just using the Newtonian equations, and you assign each atom a certain velocity. And then after some time, you look at how far out the atoms have moved, right? And you keep on repeating this process. And as a result of that, you will end up producing the movies that I showed you earlier. Now, coming back to docking. So we looked at this before, and we saw that this gray cloud representation I told you was the particular protein surface which had this cavity. And then you are trying to plug and play with all of these ligands and see which one gives you the best score. Now, remember that when you are doing something like this, this is rigid docking. So when you're doing rigid docking, you're not allowing your protein in the surrounding to actually explore or actually move. But we know from what I've just explained that proteins are highly dynamic, right? This is one of the reasons why the answers that you get from this may or may not be always correct. Now, when you talk about how do we actually, so there is the part where you do molecular dynamic simulations, right? But docking is not, so molecular dynamic simulations is step 0. Once you have produced the trajectories, then you can do your docking calculations, right? Now, when you're talking about docking, there are several different methods to do it, right? There are very quick and dirty methods like PBSA and GBSA. And then there are very specific and highly resolved methods like free energy perturbation, right? And these methods, what they will tell you is how strongly a particular ligand binds to a particular protein, right? That is what your end goal is. Now, when you are using all of these methods, what you are actually looking at is you're compromising on accuracy in the favor for speed. Now, when you run free energy perturbations, it will take you very long to complete your calculations. And some people like me can be incredibly impatient, right? So when you're an impatient person, what you will do is you will say, okay, maybe I don't want that much accuracy. So I will compromise on accuracy and I will like to complete my results in 1 hour. Right? So then you go with something like GBSA methods, right? So these are generalized bond solvent accessible methods. So because a lot of times in literature, you will see GBSA methods. So I'm going to take the next few slides to explain this. So, sorry. Okay, I guess somebody has just unmuted their mic, but yeah, so what we are doing over here is we are actually compromising, right? So we want faster results. So we're going to give up on certain degrees of accuracy, right? So one way in which GBSA methods work is, now I know this might be overwhelming to some people, but let me break it down. This delta G binding is your end goal. You want to find out what is the free energy of binding, right? Now, when you're going to be doing this, what you basically want, how will you get this? You will calculate the energy of the complex. You will calculate the energy of the protein. You will calculate the energy of the ligand. Now, why are you doing these three? Now, imagine that if you have the drug molecule or the ligand, which is shown in green here, and the protein, which is shown in red, and the water around it shown here in this blue, when you calculate the energy of green alone, and when you calculate the energy of the red alone, and then when you calculate them in a complex, and then if you take this energy, and which is, so when I use the word complex, I mean that they are in a bound state. When I talk about a protein, I talk about this alone. And when I talk about a ligand, I'm talking about this alone. So when you do complex energy minus protein minus ligand, you will end up with what is the change that happens as a result of binding. If after binding, your energy values are higher, actually they're considered to be, if they're above zero, what is happening is that your complex is unstable, right? And that means that this thing will not bind to it, right? But if the value of this delta G term is negative or below zero, what is going to happen is that this state where A and B are in a complex form is more stable than A alone and B alone. So if A alone and B alone are in a high state of energy, then just for the sake of thermodynamics, they would like to minimize their energies and would like to stick together, right? And this basic understanding is what you are trying to recover from using this equation. Now, when you talk about this equation, right, when you're talking about this, the components of this can be broken down in such a way that you can have a heat change of your system or enthalpy. You will have the solvation related. So what is the change in surface area? So remember that, for example, when there is no drug bound here, this area is exposed. But when the green thing comes and binds here, it reduces the surface area both for itself and the surface area of the protein, right? So it is no longer exposed to water. Now, as a result of that, you will have a change in the solvent accessible component of your system. And then this minus T delta S is basically the entropic contribution. And one of the approximations that people do is that because entropy is very difficult to calculate, what people end up doing is they end up ignoring this, right? So they will say, oh, because this is too difficult to calculate. So there is something called normal mode analysis, which you can do to calculate these, but people usually just ignore them. So this will get ignored. And this solvation related component can then be decomposed into the generalized Born calculation, the solvent accessible calculation, and MM. So MM basically stands for molecular mechanics. And this is where I would like to remind you that you will have to go back to the energy function that I showed you, where you will be looking at your molecular mechanics based energetic contributions, right? So you will have the non-bonded potential between your ligand and your protein, and you'll be doing that. Now, you don't have to do any of this yourself. All you have to do is to first do 3 simulations, for example. You will first do an MD simulation of the green molecule alone. Then you will do a simulation of your red sphere alone. And then you will do a simulation where the green is bound to the red. I might have forgotten to mention something, but remember that you always start with a bound state. You do not start the simulation by putting the green here, for example. If the green is here and the red is here, in MD simulations, the green might go this way, or it might go some other way, right? It might go this way, this way, this way. It may never come and bind here. So to reduce our time for running MD simulations, what we do is that we start off by having a complex. And if the complex is not correct, it will be reflected in the delta G values, right? So again, you do these 3 simulations and then you run your, so when you're doing simulations, and you will see later on today as well, when somebody teaches you about molecular dynamics, that each of the movie that you saw is actually comprised of different frames, right? So you run your calculations on each frame, and then you average it over the entire length of the movie, or as it is called, a trajectory, right? So you will do the same thing for complex, for protein, for ligand, and then you will get 3 values, and then you will subtract them, and you will get the delta G of binding. Now, that is how docking is done in using molecular dynamic simulations. But one thing that people forget to do is to remember that when you have a free drug ligand in your system, and you have a protein target, what you are expecting is that the drug will bind to the particular protein. But remember that this should be considered as a reversible process, right? Because, well, it actually depends on which disease you are targeting. In some cases, you just want to get rid of the protein, right? In certain other cases, you want the protein to be there, but maybe function half of the time, right? So maybe not let it function 1 hour every day. You might want to make it function 30 minutes a day. So for the remainder of the 30 minutes, you want to block that binding pocket, right? So in that case, your process must be reversible. Now, what does that mean? That means that when people do these calculations, they think that delta G as low as possible, like minus 1,000 or minus 5,000 or minus 50,000 or minus 100,000. Actually, they never reach those values. I'm just giving you an example that those values are going to be better, right? But the problem is that you have to bear in mind that if it is binding very strongly, it might not be willing to come off. So you have to come up with the correct balance of this. Now, remember that we are talking about delta G values, right? Or binding values. Now, when you go into a wet lab, you will not necessarily be working with delta G values. In the lab, what you see is on-off rate. So you have the K on and the K off, right? So you will have to do some kind of conversion between your delta G and your, I mean, most of the time, people who do computational work only do computational work and they don't actually go into the lab at all, right? So in that case, maybe you don't want to convert delta G into K on off. But sometimes it happens that in order to test whether your answer is correct, you might want to match it to experimental calculations. If you want to do that, then the experimental people would have given you K on off, and then you need to convert those into some proxy which can be then correlated with your delta G values. Now, why am I telling you about this reversible process? Because unbinding was previously not studied. So I'm just giving you an example of 3 papers here, but there is now an increasing interest to see how, so yes, there is an interest obviously that how does a drug bind, but there is also an interest in how the drug unbinds, right? So when you're doing your docking studies, I would recommend that you always keep this thing in mind, that what is your system that you're studying? Do you want to completely neutralize? Like, for example, in the case of the coronavirus, there, if you find something, if you find a drug that can bind to the spike protein, you want that to stay bound forever. You don't want it to come off, right? But in any other case, for example, maybe your system is over-processing something or over-producing something. So then in that case, you may want to neutralize half of the proteins present and keep the rest of the half working. I'm just making this hypothetical case up. So in those cases, you would be interested in making the ligand both bind and unbind. So I'm touching the hour mark. I'm glad I was able to finish this content. I know this is a very quick and dirty introduction to both MD and docking, but I hope you have some takeaways. My contact address would be available with the organizers. They might have already distributed it, or you can ask from them. And if you have questions now, you can obviously ask. And if you have questions later on, you can always come back to me over emails, and I'll be happy to take your questions. So I'll open the floor to questions now. If somebody has any questions, you're welcome to ask. If you don't have questions, then I will turn this over back to the organizers, because that is all I had to talk about today. So thank you everyone for joining. And are there any questions?
Okay, thank you very much, Dr. Ashar Malik. My name is Mohamed Ahmed Jallo. I'm a student at Institute Pertanian Bogor University, master's student in biochemistry. Actually, I'm not going to ask a question. I'm part of the committee, and I'll be moderating the question and answer session.
Sure.
But first of all, we would like to appreciate you for the extensive and detailed presentation. Although, as you mentioned, due to the time constraints, we cannot go into detail to some of the concepts and methods. But I think this is a very, very good and brief and detailed introduction to molecular dynamic simulations. So while we are waiting for questions from, I just want to remind the students that there are two ways you can ask questions as usual. You can ask your questions by leaving a message in the chat room where we're going to read these questions out. Or you can ask your questions by raising your hand, wherein you can ask your questions verbally by voice, so that Dr. Ashar Malik can respond to your questions, all of your burning questions you have for molecular dynamic simulations, most especially using plant materials. So we're gonna give it some time to check this.
I mean, people are usually shy, so you don't have to be shy. You can openly ask any question if you want. If something isn't clear, please feel free to ask.
Dr. Malik, if I may ask, I don't know if it's okay if you can turn on your camera and
sure.
stop sharing the screen. That will be very well appreciated. Turn. Okay, thank you very much. Let me.
So I think Umar has raised his hand.
Yes. Okay. Thank you. I think we have someone raising their hand. Okay. Mr. Setianto Umar, you're welcome.
Thank you. I have first one question about the calculation with using the PBSA, Poisson-Boltzmann surface area, and or GBSA. Usually the calculation using the entropy is the hard part to calculate, because the entropy is too difficult to calculate and computationally costly. Is it possible, or is it common if we just calculate only the enthalpy part of the GBSA?
So, okay, so let me clarify this. When I said that it is difficult, what I meant was that it is only time consuming. The reason why we don't want to use the word difficult is because nobody's doing this on pen and paper anymore, right? So back in the 30s and 40s, people used to actually run molecular dynamics using calculations which they used to solve on a piece of pen with a pen and paper, right? So at that time, the entropic calculations were difficult to do because it required a lot of maths. Now what happens is that there is just one plugin and you just have to write 4 lines of code, and then the computer will actually calculate it. So it's not difficult, right? The only part is that it is time consuming. And the reason why people tend to ignore this is that after you run your entropic calculations, the value is so small that if you would not have spent 2 weeks calculating that and had just ignored, it would not have affected the answer that you would have gotten by only solving the enthalpy part, right? But again, this has to be taken with some salt, because remember that not all proteins are identical, right? Every protein, the way it functions is different. And in some cases, it might be that the enthalpy is not driving the conformational change of how the protein opens up and binds. It might be entropy that is actually the driving factor, right? You just have to be careful about your own system and how the changes that are happening in your system can be accounted for. I hope that addresses your question.
Okay, thank you.
You're welcome.
Okay, thank you very much, Mr. Setianto Umar. We hope that answer was, as Dr. Malik said, helpful. Guys, we are still waiting for your questions. Dr. Malik is here to answer all of your questions or some of the things that you didn't get clearly when he was explaining. So I think this is the best time to have your questions answered and your things you have misconceptions. I too had a molecular bioinformatics course last time, this gone semester. Although I'm not a bioinformatician, but from there on to this time, with the addition of this lecture, I've had I usually had a misconception of the issue of molecular dynamic or molecular docking, but like after this lecture, I've had some level of clarity on how what's the exact definition, what's the exact meaning of this issue, because there's a lot of misinterpretation, especially about the functional activities of these things. So while we are waiting for some questions, we'd like to, because we know most of the students here, due to the pandemics, a lot of people have difficulties working in wet labs. So as a result, many people are resorting to the use of bioinformatics, downloading data from the NCBI and that of the Protein Data Bank or UniProt to do bioinformatic research. So I would like to know, on behalf of the students, would like to know what are some of the challenges for a startup, for someone who is just going into this field of bioinformatics, most especially for master's students or undergraduate students. How does it feel like starting up in this area of research? What are some of the challenges to be expected, and how can we overcome these challenges?
Okay, so so before I answer that question, I just wanted to say that I have received another question directly in my in my chat. So please remind me to answer that so that I don't forget. Okay, so there is no right or wrong answer to your question. So I'm just going to give you my explanation about what is happening and how what are the challenges, right? I think one of the biggest hurdles is not understanding the system that you're working with and not understanding the limitations of the tools that you are using to explore your system, right? So I think this is one of the biggest challenges, because what people, so you might have heard that 90%, almost 90% of the published literature out there actually comprises of non-reproducible results, right? So people, either the results are not translatable in the lab, or people are not able to actually produce accurate results, right? So the primary reason for this to happen is that when people look at a paper and they say, oh, look, I can do step 1, step 2, step 3, step 4, and I will get my answer, right? And then they don't understand why. I mean, there's a chance that the person who published that paper might have been following somebody else's protocol.
Yes.
Right. So, incorrect publications leading to incorrect publications is one of the primary factors why people continue to produce results which are not significant. They take up a lot of your time. They take up a lot of taxpayers' money. They take up a lot of other logistical resources, but they do not end up translating in a meaningful way to the progression of the field. So if you ask me directly, what is the one challenge that people can, focus on, it would be to first understand the tool that you're using. What was it designed for? What are its strengths and weaknesses? People don't do that, right? Then understand the method that you are actually using. So if you're using MD, don't just take a protein structure and run a standard MD, because there is a chance it might work, but there are a lot of things that you have to do to your structure to prepare them. A structure cannot be directly taken from RCSP and simulated. You need to do a lot of both major and minor changes to the structure before it reaches that stage that you can actually run a simulation on it, right? So just don't black box. Don't take a tool put something in and expect something out. Because if junk goes in, junk will come out. So
yeah, obviously.
yeah, exactly. So I think if you ask one answer to your question, it would be to just understand your system, understand the tool, understand the methodology, and then apply so that you understand the limitations and that the conclusions that you make in your paper are then based on what are the limitations of the tools that you've used. Okay, so I hope that summarizes your answer. I know this is a very philosophical question, so I don't want to spend too much time.
Yes. So let's get back to the question you have on your private message board.
So the question that I got was from Al Farizi, and the question is that they're talking about reversible and irreversible binding, and if that is the reason why paracetamol, was it paracetamol? Paracetamol can cause hepatotoxicity. So, okay, so the first thing is I don't know the exact mechanism on how paracetamol causes hepatotoxicity. But what I can comment on is that when you develop drugs, now paracetamol is a very established drug. It has been around for 50 years. But let's suppose that you're talking about a new drug, right? There is something called off-target effects or off-site effects or non-specific interactions. So what does that mean? That means that if you take drug A and if you take protein A and you want this drug to interact with protein A, remember that the cell is a very packed system. So when there is protein A, just around it is protein B, just at top of it is protein C, protein D, protein E, and this cell is full of proteins, right? So there is a chance that the drug will also interact with other proteins, right? This is called off-site effect. So on-site is the effect that you want on protein A, and off-site is when the drug starts and goes and interacts with protein X or protein Z, right? Now, to address your question, if there is a chance of paracetamol, I don't know if there is, but one of the pathways is that because paracetamol gets absorbed in the blood, right? And it goes systemically to all parts of your body, right? So naturally, when the blood goes to the liver, there is a chance that, obviously the liver does detoxify. So the liver role is to actually clean up the blood, right? So it might be that there is some interaction where the paracetamol can have some off-site effect. But I know people who have taken paracetamol regularly for 20 years and there is no damage to their livers, right? So I think it might be a case-to-case variation, or it might be that certain people that you are talking about may have certain mutations in their genomes, which makes them more susceptible to in having these off-site effects. Again, there are a lot of factors. When you study computationally, you expose one drug to one protein, right? And that helps when you are in silico, but that is why you go from there to lab in vitro, and then you go to a mouse model, and then you go to a chimp model, and then you go to clinical trials, and then you roll out the drug. Because in all of these steps, you are trying to find out, is there anything that you have missed? Is there any off-site effect which might be so big that it might, you might be taking it for your headache, the paracetamol, but if it kills your liver, then you better live with the headache than without a liver, right? So you have to strike the right kind of balance between what are the good effects of the drug and what are the side effects. So I know that this did not exactly answer your question, but I hope that it gives you some points to think about when you look at drug interactions with proteins that they're not supposed to be for. So yeah, I know you asked about reversible, irreversible, but because I don't know the mechanism of how hepatotoxicity is caused, I can't really comment on it.
Okay. See, still have a little bit of time to go.
You're welcome.
we still have some time, so probably we'll generalize some questions, and because I'm not the student, so we'll generalize some questions. I mean, while we are still waiting for our students to come with specific questions related to the topic, maybe for my questions will be on a general basis on the dummy level for students interested in field of bioinformatics for research. So today we are talking about, probably we have a question here.
Yeah.
From Fashrul Rizal Mahendra. Unfortunately, Fashrul did not mention the institution he comes from in the group. Please, it will be very, very good if all of the participants will include their group and that of their name. I mean, the institution they come from. So Fashrul Rizal, please, would like an additional comment of your institution, that is your home university or your affiliation. So let's get to the question. Let's get to the question. I don't know. Sadia,
no, I can answer. I can read out the question. So the question is basically talking about how do QM/MM in MD, and what MD tool can do QM/MM for enzymes. Okay, so as I said before, that when you're doing molecular dynamics, you can't do QM, right? So QM, there are certain tools that are designed only for QM. So for example, there is DFT theory, and there are programs like the Gaussian software. I think it's called the Gaussian. I actually have not used it, so I am not familiar with the quantum level calculations. But there is DFT theory, and there is a program called Gaussian, right? So when you talk about metabolic reactions, right, or when you talk about anything where a covalent bond will break and new bonds will form between different atoms, that is what happens in a metabolic reaction, right? So you, for example, if you have glucose breaking down into carbon dioxide and water, what is happening is that there is bond rearrangement. So in molecular dynamics, you cannot have a bond rearrangement, right? So if your system starts off as a protein which has 10,000 bonds, it must end with a protein, the exact same protein, exactly having 10,000 bonds, right? So there can't be any rearrangements. The only thing that can happen is non-bonded interaction. So we talked about how you can approximate using those energy functions. You can approximate that when a ligand or a drug comes together with a protein, it will start interacting using hydrogen bonds. Now, hydrogen bonds are not real bonds. They are derived because of the charge differences, right? So you have positive, you have donors and acceptors, and based on their proximity, you have interactions that develop, right? So the important thing to remember here is that when you talk about MM or molecular mechanics, what you are basically asking is how does a molecule do what it is doing, right? So that is the underlying definition of molecular mechanics. And to understand that, you will then use either the energy function that I showed, which was charm based, if you're using charm tools. So you can use charm force field with GROMACS as well, or NAMD. But when you talk about enzymatic calculations, if you have the world's fastest computer, for example, and if you have maybe 10 or 20 of those, for example, you may be able to simulate the full protein coming around, attaching to a particular metabolite and breaking it up, right? So this is an enzymatic reaction, right? So a protein, like for example, the protein that breaks down glucose. So in the glycolysis pathway, for example, there are a lot of proteins, right? And those proteins are responsible for actually taking glucose and breaking it down, right? So when you have to simulate those kinds of reactions and you want to do quantum level calculations, you have to have a lot of time. Now, there are tools which do QMMM, but they are only approximations. I am not familiar with those, but I have read about them in the literature. So you can use those, but they will still not be able to reproduce what you are asking. So if you want to do a full QM level calculation on an enzyme to see its metabolic pathway, you will need a very fast computer, and you will need to spend a lot of time actually doing that. So some of the theory that you will use is DFT, not Newtonian equations, like we talked about in molecular dynamics. And you will have to you will have to obviously spend a lot of time into the exact region of the protein where the reaction is taking place. So I hope that addresses your question in some way.
Yeah, hopefully. We we had someone raised their hand for a question. I think Sadia,
Sure.
but I think the hand was dropped again. So if you are there, please come in. You can unmute yourself and probably turn on your camera for your question. Sadia, I don't know if you're still around. So while we are waiting for Sadia for her question,
You're welcome.
Okay. I just heard you talking about fast simulations. We know that time in molecular dynamics simulation is very, very of essence, but not just time, because during your presentation as well, you mentioned that there are sometimes people will opt for not so much accuracy, but instead they'll just want to get their stuff done in time. So how does AlphaFold, I know you might have heard about AlphaFold, this Google AI, how does it factor in this situation? What future does it have in the area of bioinformatics or you know, like whatever it is going to be doing in this future of molecular dynamics, bioinformatics, or artificial intelligence? How does it factor in this protein modulation?
So, okay, so I don't have a correct answer to that. So we have limited exposure to AlphaFold as of present. I've only seen very limited results from it. So let's talk about what AlphaFold does. Okay, so AlphaFold is not a molecular dynamics tool, right? What it does is it uses a certain AI principles to actually predict structures given sequences. I mean, that's a very, very crude definition of what AlphaFold does, right? So there were tools before AlphaFold as well, right? So every year there is a competition called CASP, where people go and bring their own methods and they compete to predict the structures of proteins. So there is an experimental body. They crystallize protein structures and then they don't publish it, but they give the sequences of those structures to these people who are competing, and they predict their structures. But because they already have those structures experimentally determined, they actually match those structures to see which prediction algorithm is working best. Okay. So this has been happening before AlphaFold as well. And there were every year some team wins, and every year what happens is that on certain proteins, certain algorithms work and certain others don't work, right? So one algorithm might be able to predict very accurately on protein A, but might fail on protein B, then might work 50% on protein C and 25% on protein D. So this is a varying level of output. Now, those things have been thoroughly tested in the scientific literature, and we have a lot of knowledge about them, right? AlphaFold is at a very early stage. What they are claiming is that their system does the job very well, and maybe it does. And the reason why it would is because instead of trying to understand how proteins fold, that was the question that we were dealing with for the past 60, 70 years, right? So how does a protein go from a linear chain to the final three-dimensional structure, right? That is the protein folding question. So now what we have started doing is that we have stopped trying to figure out the answer to that question. And what we are saying is that we now have in the PDB database, we have, let's say, 500,000 individual protein structures, right? So if we have so much structures, maybe we can leverage on the AI methods, machine learning, deep learning, other kinds of approaches to this, and that we can, instead of predicting how the protein is folding, we can use this knowledge to actually create a black box function where, without understanding, our mathematical models can take in the input sequence, and based on their learning from these 500,000 structures, they can give out a structure, right? Now, there are obviously problems with this approach, and one of the problems is that if your model is based on junk, for example, which in this case it is not, it is based on very accurate 500,000 structures. But I'm just saying that if your model is based on junk, obviously it will give predictions of junk, right? Now, why am I using the word junk here is because if you talk about protein structures, you are heavily based in homology. And you say that if homology is present, then you are going to use homology to predict the structures, right? So the assumption is that proteins that have the same amino acid sequence or similar amino acid sequence will fold into three-dimensional structures that look similar, right? That is the underlying assumption. And we know from before that information is not 100% accurate. I gave the examples in the beginning where you may have even up to 80% similarity at the sequence level, but the structures might look completely different. Or you may have as little as 20% sequence similarity and the structures might look exactly similar, right? Not exactly similar, they might look similar, right? So again, coming back to your question about AlphaFold, we at present, what we can do is that we can take those predictions, we can feed them into MD simulations and see how MD behaves. But again, if you junk into MD, junk will come out of MD as well, right? So AlphaFold is at a very early stage. What we want to do is that we want to take its predictions and we want to verify whether those predictions are correct or not. And as the scientific community's confidence in AlphaFold builds up, it will become a more established tool. So at present, we won't be able to address, I won't be able to say more on AlphaFold. It's a good tool. It's a promising development, and hopefully it will answer our questions. So that's all I can say.
Yes, I think you covered the question, not just directly, but, you know, with the surrounding misconceptions that are associated with the question as well in AlphaFold. So thank you very much, Dr. Ashar Malik. We've seen our MC is already here. It's a sign that our time is limited. So thank you very much. We really appreciate you being here with us today and all the insightful information shared. We believe that the students will take advantage of this information and venture into the research field of bioinformatics, most especially molecular dynamics, because we have seen how important this field is, not just most especially with the recent advancement in research and the obvious therapeutic research, most especially for COVID-19. So thank you very much, Dr. Ashar Malik, and we are glad to have you. And for the students, if you have follow-up questions for Dr. Ashar Malik, please get to the committee. They will make sure your questions reach to Dr. Ashar Malik, and then you'll have